the second series of the lecture, the second one, cooperation between innate and adaptive immune responses, innate and adaptive. Last video, in the first part of it, we had given an introduction, what is innate, what is adaptive. Innate, the first line of defense, barrier function, supplemented by certain methods of killing, for example, by natural killer cell, phagocytes like macrophages, then some proteins like interferon, complement, and processes, inflammatory processes. But the innate lacks certain very important points. It not specific, adaptive is very highly specific. And then adaptive has got certain other advantages also. It is diverse, self, non-self discrimination, memory, etc. So those advantages which are possessed by the adaptive immune system, they are put into use for innate also. As a result, the overall, the synchronized action of the two becomes very effective. We are going to see with the help of three examples. Please note, the first one is ADCC, antibody-dependent cell-mediated cytolysis. Antibody-dependent cell-mediated cytolysis. The two soldiers in this cooperative endeavor are antibodies, component of the adaptive response, cell-mediated, and the cell here is natural killer cell, a component, a soldier of the innate immune response. So we are going to see how that happens. Now just visualize a pathogen entering the body. So a pathogen, it has breached the barrier, the skin barrier of the innate immune response को ये breach करके it has entered the body. Now it will be first countered by the natural killer cells, among others, natural killer cells. But here is a disadvantage natural killer cells they have. They do not exhibit any high degree of specificity for the invading pathogen, and here comes into play the cooperative action of the immunoglobulin. Immunoglobulin, through its FAB site, recognizes an epitope on the invading pathogen, and the other site, FC site of the immunoglobulin, इसके receptors होते हैं natural killer cell पर, it binds here, and in this way you can see that immunoglobulin is acting as a bridge between the two. And now natural killer cells, the field is clear for them. They will secrete certain proteins, perforins, which dig hole into the invading pathogen cell wall, and then certain granazymes, which enter the cell through these pores, and then they will blast, they will destroy the pathogen. So that is first we have seen ADCC. Second one, complement, very simple now to understand for you. Again, a pathogen is entering और इसको काउंटर किया antibodies ने through its FAB site, and then this is the FC site of the antibody. In fact, see the word, it comes from complement. So FC site, it will bind complement protein. Complement proteins are a group of about 30 proteins or more now discovered, which act in a self-amplifying manner, a cascade-like action you are very well aware of. As a result, the effect becomes very much amplified. The first one to bind is C1. Then after that, C2, C3, C4, C4 till C9, they get activated, not in that order, by numerical order, of course, but ultimately, all the complement proteins, they are getting activated, and together they are going to form a complex which we refer to as, abbreviate as, membrane attack complex, comprising of various complement proteins, and membrane attack complex will dig holes into the invading pathogen bacteria के cell wall के अंदर. So that way it is destroyed. Complements, they have got another role to play. This complement protein, they have got chemotactic action. Chemotaxis, that means they recruit various other cells to the site of action. Neutrophil, macrophages, etc. इन सबको भी यहाँ पर attract कर लेते हैं, so that the effect gets amplified several folds. So these two are there. Now we are coming to the third one, that is opsonization. Opsonization, ये invading pathogen usual immunoglobulin ने किया FC site. FC site, it has got certain receptors. I mean, these are phagocytotic cells now coming into action. Macrophage. अब macrophage के ऊपर there are receptors for this FC portion of the antibody. So यहाँ पर fixing हो गई. So as a result, pathogen and phagocytotic cells, they have come in close proximity. I have not shown this figure in scale actually. Scale से अगर हम देखें तो you can see here that the pathogen coated by immunoglobulins. Immunoglobulins are very small. So I have shown only one, but so many immunoglobulin bind and coat it. And then macrophage, very easy for macrophage. So coating of the pathogen by the immunoglobulin makes the phagocytotic process very easy. So this process is called opsonization. I don't know the literary meaning, putting on the table or something, that immunoglobulins are presenting in a very presentable way the invading pathogen to the phagocytotic cell. One last point, complement proteins also, they participate in process of opsonization. They also coat the invading bacteria. So these are various, I have talked about the three methods. There are some more also mediated by cytokines or T helper cell. We are going to talk in the next video, which is going to follow very shortly. Thank you so much.